Hello everyone, a very warm and pleasant welcomes to you all. Today I, Sweta Thakur, the student of Dr. Hari Singh Gaur Central University, is going to present my presentation on a topic basic structure and properties of cytokines. So before proceeding towards my topic, let me tell you what are cytokines. Cytokines or immunocytokines is derived from a Greek word cyto and kinos. Cyto, which means cell, and kinos, which means movement. They are low molecular weight regulatory proteins or glycoproteins secreted by white blood cells and various other cells in the body in response to a number of stimuli. They role in cell to cell communication. They help in transmitting signal from one cell to the other. The development of a strong and effective immune responses involve some type of cells like lymphoid cells, inflammatory cells, and hematopoietic cells. Hematopoietic cells like white blood cell, red blood cell, and many others. Now, let's see the definition of cytokines and some terms related to it. Cytokines can be defined as a group of proteins made by the immune system that act as chemical messenger in transmitting signals from one cell to the other. It is the general term for a large group of molecules which is involved in transmitting signal between the cells during immune responses. Cytokines are generated by mononuclear phagocytes, which are known as monokines, and those which are activated by lymphocytes are known as lymphokines. Additionally, both monocytes and macrophages produce cytokines such as CSF, colony stimulating factor, which stimulates or help in the growth of immature lymphocytes in the bone marrow. Now let's see some basic properties of a cytokines. All cytokines are proteins, some with sugar molecules attached known as glycoproteins. They are synthesized and released by the white blood cells and tissue macrophages. So, white blood cells are formed in the bone marrow. And what is macrophages? Macrophages are specialized cells which is involved in phagocytosis. Cytokines stimulate or suppress the functional activity of lymphocytes, monocytes, neutrophils, fibroblasts, and endothelial cells. There are 200 different cytokines have been identified. Our next properties are cytokines bind to specific receptors which is present on the membrane of target cells. So we can say that cytokines are target specific. They are long known to be involved in cellular immune response. These products have additional effect that they play important role in acute and chronic inflammation. So cytokines helps in cellular immune response as well as they help in acute and chronic inflammation. Various cytokines mediate their effect or various cytokines show their effect by binding to specific receptors which are present on target cells. And thus expression of these cytokine receptors can be regulated via a variety of exogenous as well as by endogenous pathway. Cytokines may be proteins, peptides, or maybe in glycoproteinous in nature. Cytokine receptors may be made up of from several different genes. Cytokine regulate the intensity and duration of immune responses. Now let's see how cytokines initiate cell-to-cell signaling or how they transmit signal from one cell to other. In this diagram, we can clearly see that the cytokine producing cell when get stimulated releases the cytokine which then get activated, which then get attached to the membrane of a target cell, which then activates the target cell for gene expression, which will then have some biological effects. Our next property is affinity. Cytokines and their receptors are highly specific and they exhibit high affinity for each other and delivers intracellular signals. So if the receptors are not present on the surface of cell, then the cytokines will not induce signal and the cells will not get activated. So the presence of receptors on the membrane of a target cell are very important. The bonding or affinity between cytokine and receptor is strong with dissociation constant ranging from 10 raised to the power 10 to 10 raised to the power 12 molar. Cytokines and their receptors show biological effects at a picomolar concentration. So here picomolar concentration means it is the non-SI unit of molar concentration. So if the cytokine is present even in a very small amount, then also it will activate the cell and the cell will have some biological effect. Now let's see the action of cytokines. Cytokines may act in three ways. So a particular cytokine will possess only a one mode of action. There are three modes in which cytokines act. First one is autocrine action, second one is paracrine action, and the last one is endocrine action. First, let's see the first one, or that is autocrine action. In this action, a particular cytokine may bind to the receptors present on the membrane of the same cell through which it gets secreted or through which it gets produced. When the cytokine acts on the same cell which is producing it, then it is known as autocrine action. Our second one is paracrine action. In this particular, cytokine may bind to receptor present on the target cell in close proximity to the producer cell. So, when the cytokine binds to the receptor present on the target cell which is present in close proximity, or when cytokine bind to the receptor which is present in the nearby cell, then it is known as paracrine action. Our last one is endocrine action. In few cases, it may bind to target cell in distant part of the body. So here in this diagram, we can see that the secretion is at one place and the receptor is present at the distant or at far place. So in three ways cytokines can bind to receptors. Now let's see the types of cytokines. The major cytokines include interleukins, that is IL, lymphokines, monokines, interferons, that is IFN, colony stimulating factor CSF, chemokines, tumor necrosis factors, that is alpha and beta. So first let's see the interleukins. They act as mediators between leukocytes. They help or they transmit the signals between the different leukocytes. The vast majority of these are produced by T helper cells. Lymphokines, they are produced by lymphocytes. Monokines, they are produced exclusively by monocytes. Interferons, they are involved in antiviral responses. CSF or colony stimulating factor, as I have told before, they help or they support the growth of blood cells. Now let's see some basic functions of cytokines. Our first function is they stimulate development of cellular and humoral immune response. Because they help in T cell and B cell activation and proliferation, so we can say that it helps in the development of cellular and humoral immune response. Second function is induction of inflammatory response. There are many cytokines which helps in inflammatory response as well as in intra-inflammatory responses like interleukin 1 and TNF. A third function is regulation of hematopoiesis. We can see that there are many cytokines like macrophage, CSF, interleukin 13, interleukin 12. These all mainly support and induce hematopoiesis. They help in formation of hematopoietic stem cells like white blood cell, red blood cell, B cell and T cells. Next function is they act on the bone marrow to stimulate or to support the growth and differentiation of lymphocytes as well as leukocytes. They, con- they help in control of cellular proliferation and differentiation. There are major cytokines which help in proliferation and differentiation of B and T cells. For example, like interferon help in B cell proliferation and differentiation. Our last function is healing of wounds. Healing of wounds is done by chemokines, which means movement of chemical towards the injury, and this is done by macrophage inflammatory protein 1 and interleukin 8. Our next function is cytokines can activate many cells. For example, cytokines secreted by T helper cells can affect B cells, macrophages, NK cells, that is natural killer cells. Cytokines can be redundant. Redundant means when two or more cytokines that mediate similar functions are said to be redundant, or when different cytokines can have same effect, then it is said to be redundant. So here in this diagram we can see that when interleukin 2, interleukin 4 and interleukin 5 act on the B cell, that is our target cell, all of them three will show same effect, that is proliferation on the B cell. So we can say that cytokines can be redundant. Our next function is a cytokine can synergize with each other. Cytokine synergies occurs when the combined effect of two cytokines on cellular activity is greater than the additive effects of the individual cytokines. So we can say that cytokines are also adaptive in nature. Our next function is our cytokines can antagonize with each other. Here in this diagram we can see that when the effect of one cytokine inhibit or offset the effect of another cytokines, then the cytokines get antagonized with each other. Our next function is our cytokines can be pleiotropic. When a cytokines may have different biological effects on different target cells, then it is said to be pleiotropic in nature. In simple words, it means one cytokines can have different effect on different cells. So here in the diagram we can see that when the activated T helper cell secretes the cytokine named interleukin 4, then the interleukin 4 will have different effect on different target cells. Like interleukin 4 when affects the target cell, that is B cell, it will show the effect like activation, proliferation and differentiation. When interleukin 4 will act on the target cell, that is thymocyte, it will affect the its proliferation. And interleukin 4 when affects on the mast cell, it will show proliferation. So here we can clearly see one cytokine can have different effects in different cells. Our next function is cascade effect. Cascade effect can stimulate the production of other cytokines. Cascade effect occurs or cascade induction occurs when the action of one cytokine on a target cell induces that cell to produce one or more other cytokines, which in turn may induce either or other target cells to produce other cytokines. Here in this diagram we can see that when the activated T helper cell get activated, then it secretes interferon gamma, which further secretes macrophage which further secretes interleukin 12, and interleukin 12 activates the or further activates the activated helper cell. This means one one will have the activation of T cells by B and T cells and the antibody combination, then which further gives out interferon gamma and interleukin 12, and this together secretes interferon gamma, interleukin 2, and maybe depending on infection some other kind of cytokines can show cascade effect. So at last, we concluded that the cytokines are a group of proteins that are involved in normal and pathologic physiological functions. They are primarily thought of as mediators of immune and inflammatory responses, but they may also play their role in growth and differentiation of other cells. Like we have seen, cytokines helps in the proliferation and differentiation of B and T cells. Cytokines may influence the action of other cytokines. Like hormones which they resemble, cytokines act by binding to receptors on target cell. The same cytokines may be made by different cell types, and individual cytokine may act on more than one cell type. I hope you all might get understood. Thank you.